Tēnā koutou, tēnā koutou, tēnā koutou katoa. Ko ka pukatama haka te moanga, ko pakahakia te awa, ko Robert Henderson te waka, Anari e tipu, aki oki o te poti, ko Brian toka papa, ko Kay toka mama, ko Janet hoa wahine, ko Stephen Rato, ko Jeffrey, ko Richard, aku tamariki, ko Gary Evans toko e ngā mi nui kia koutou. Hi, I'm Gary. I'm originally from Dunedin. I really shouldn't be chewing up time, limited time to talk about my career in what will be 9 minutes probably, but I think it's really important. I mean, it's who I am, where I'm from. My parents are here today, and it's a fantastic opportunity just to tell them what I do, because I don't often do that. So today I'm going to talk a bit about finding the right chemistry. I mean this in both a descriptive and a figurative sense. I am a chemist. I make molecules, I make drugs, and I don't taste them, Owen. I haven't tasted them recently, put it that way. But it's all about finding the right chemistry and working in large teams. And I think some people have talked about this today. Research at scale is how you really create a lot of good impact. And I suppose finding the chemistry within the team, finding the right people is really, really important. I've tried to avoid words as such, so I'm going to start out with a sponge, because I really got into chemistry cooking beside my mother, who taught me how to bake. My father extracted linseed oil from linseed, and I used to hang around the factory there and when I was very young, and I loved the smells, loved the gear. And so when I went to university, to secondary school, I had some really good chemistry teachers, and that I think combined threw me into a career to do with chemistry. Went to the University of Otago, and funnily enough, one of the honorary fellows today is the son of this man, Peter Grant, who was my mentor at university. I did my PhD with him. I didn't know this before I read the booklet this morning, but he was fantastic. He was known as the bionic arm, and he taught me the fundamentals of what it was to be a really good researcher, and a lot about chemistry. And that whole late 1980s group at the chemistry department at the University of Otago taught me a lot. From there, I was able to go out and work in this kind of area of organic synthesis and stereochemistry at the University of Oxford. I wasn't as impressed with Oxford as I thought I might be, because the gear wasn't all that flash. The facilities weren't great, but there were great people. I mean, it was chock with great people. And this fellow here, as well as having an impressive pair of mutton chops, was very entrepreneurial, and he taught me a lot about chemistry, about being an entrepreneur, although I can't claim to really have taken on the lesson particularly well. He remortgaged his house to set up a company, doing some of the chemistry that's shown there, and was able to form a company that he sold for, I think, £321 million about 5 years after I left. But in that role, I learned a lot about doing what I did at the bench on a small scale, millilitres, doing it in tens of litres, hundreds of litres. And so there was a whole lot of skills there I was able to bring back to New Zealand. And at Industrial Research Limited, with Richard Furneaux and the team, through a Foundation Research Science and Technology postdoctoral fellowship, and there was able to build up a process development group, able to put together a business case for a chemical synthesis facility, which is now Glycosin and sits out at Callaghan Innovation, where I worked for 1 year. And so it's a fantastic facility, and something that I really enjoyed doing. But what I really, really enjoyed doing was research and drugs, coming up with drugs to treat different diseases. And we had a fantastic collaborator. Here we are. Probably the two most important influences in my life, other than Vern. They've all got glasses on, so I can't really differentiate people. On the left is Peter, on the far right is Richard, and in the middle is Vern Schramm. I'm the guy standing over them, obviously. And so through them, I learned a lot about research, about trying to find uses or impact through what I did as a chemist. And this was a collaboration between what was Industrial Research Limited at the time and the Albert Einstein College of Medicine. Vern had drawn us a structure to make. Peter went away and made something very similar to that. I'll talk about what I did a bit later in making the actual compound itself. But we were able to license the compound that Peter made to BioCryst. These compounds were described in a series of papers alongside another compound that went into clinical trials against SARS-CoV-2. But the compound Peter made that was described in that paper actually made it into the clinic, is approved for use in Japan. And so that's what I think of as impact. I mean, that set the bar pretty high early in my career, I have to say. And so my role was, in part working alongside Peter, supporting Peter as part of a much larger team. I was able to learn a lot from him. He's certainly my role model in life. And so I came up with a new generation of these compounds that also went after a similar target, but were not far better. They were better inhibitors than those. And this was something we designed rather than Vern. And so it was something the chemists brought to the party. We were able to show the biochemists and the structural biologists that we could actually bring something to play here. And so this compound sat in his fridge for a while until Richard went and reminded him that he might want to test this compound. And then I got a call on a Saturday morning telling me, this stuff's great. And they were able to license that. We licensed a whole lot of these compounds to this small biotech called BioCryst. We licensed on to Roche. And Roche paid IRL and counting on $30 million with promises of $530 million as in milestone payments that never really came to fruition, and took it into a phase 2 clinical trial for psoriasis and made hundreds of grams of this, or kilos of this material. And so that's what I call impact too. And that led to me winning a Royal Society award, so I love the Royal Society. In terms of these compounds, what we're doing is targeting an enzyme. We're trying to stop an enzyme from functioning, and that enzyme is important in a disease process. And the compounds we made are actually what we would call the perfect enzyme inhibitors. They go into the enzyme's active site and they stay there for the lifetime of the enzyme. And so I eventually made the compound that Vern had scrawled on the bar napkin. I mean, you hear these stories about people scrawling things on bar napkins, but it really does happen. And so he'd drawn this. I eventually got around to making it. It wasn't as good as Peter's version. Peter had made something slightly different, just as complex, but slightly different. His molecule was better. The second generation compound I made was better, but it was a really, really good starting point. And this is often what we need as chemists, is just somewhere to go, somewhere to aim our skills, somewhere to point ourselves in the right direction. And these compounds all went into a series of different trials for things like gout, malaria. It actually cured malaria in a primate model, but it was going to take too long. 7 days as a cure, when the Gates Foundation, I wanted something that would take a single dose when you've got people coming into clinics in sub-Saharan Africa and then wandering off again. They wouldn't stay around for the entire course, and so it was problematic for them. They wanted something really, really simple. More latterly, I've worked with some other fantastic people like Parry Guilford at the University of Otago, who's been looking at a particular form of stomach cancer, particularly predominant in a Māori population on the east coast of the North Island, where they have a genetic mutation. And so what Parry was looking at was a model where he had a knockout mouse, and using a synthetic lethal approach, we were hoping to come up with a compound that would hit the genetic mutation and kill the cancer. And so working alongside Andreas Luxemburger, we were able to publish some work just very recently in the Journal of Medicinal Chemistry on this. And so that's been a really satisfying relationship. I just got an email from Parry yesterday wanting me to tell him how to make something. So he's just fantastic. And I think that's, again, this chemistry of being part of bigger teams across institutions and doing stuff that is excellent within a team. And so I suppose finally I get to the point where I ask, what is excellence? Another really big role model of my recent career has been Prue Williams at the Ministry of Business Innovation and Employment, where I went and worked as the Chief Science Advisor for 4.5 years. And so while trying to keep my research career on a sort of a lifeline ticking along in the background. And so Prue has been absolutely fantastic in supporting me in that role. I met some fantastic people like Hamish Spence, of RSNZ, Rod Murdoch, who's the Deputy CEO at NIWA, who worked with me as science advisors at MB. And together we put together a paper on what was excellence in research in the current context, and we're able to work with people like Wendy Lana and others in the sector to give us their views and try and reflect some of the modern approaches to what does it mean to do excellent research, particularly in an Aotearoa context. And so that was published, but it is an MB policy, unfortunately. And so more recently, I've obviously left MB a couple of months ago, and so for me the future has been working on the development of antivirals alongside Vernon Ward at the University of Otago, another rather large MB program. We've just published recently an isomer, which is just a slight variation of the compound that already made it into clinical trials that Peter had invented way back in 2000. And so that's been an interesting process. We started all this before SARS-CoV-2, and it becomes very topical all of a sudden, a lot more topical than it was when we started. I'm now in a new laboratory. Richard has, we've put in place a succession plan. I'm in charge of what is the Ferrier Research Institute, which is the group on the right. And so back at Gracefield, I've been in 3 different organisations, all on the same site, IRL, Callaghan Innovation, and now Victoria University of Wellington, and at the Ferrier, and really looking forward to the future. It should be exciting. So finally, I'd just like to acknowledge a few people, my mum and dad, brother and sister, my 2 favourite guys, Richard and Peter, and just my family from a recent 21st, you can probably tell from the beer, but in particular my wife, who's fantastic. She really has put up with a lot. So yeah, that's my career in a very short period of time. So thank you so much. Cheers.
Well, thank you for that. We can see the rocks from which you were hewn. So it was very interesting. I was also interested that you had an inspirational high school chemistry teacher. I didn't have a chemistry teacher in high school because nobody could teach chemistry, but one teacher walked in, put a book down and said, "You should read this." So what we needed was the Einstein drug, which we didn't have in those days.
Yeah.
But anyhow, does anyone have a question they'd like to ask of Gary?
Gary, you asked me the question this morning. What's been the role of IP in the work?
Yeah, it's kind of a it's a really tricky question, because I think it's evolved a lot since the noughties, when I suppose it was kind of our heyday really in terms of taking intellectual property, licensing that, and having someone come along and invest in it. And the key there was probably having people in the States who were working alongside us to try and find partners who would invest their money in our ideas and take them forward, because they're really expensive to actually take it from a concept into the clinic and forward. Eventually it happened, but that cost a lot of money. They defended an awful lot of patents on our behalf that they'd licensed as part of the families that we'd licensed to them, in all sorts of jurisdictions. I mean, that's hundreds of patents, and they're paying for it and maintaining it in order to. So it's important, but you can also see where the government's coming from, where I think they want to invest and get as much bang for their buck across the community as a whole, rather than having an individual company lock up that intellectual property that's been generated out of government investment in research. But you can't. I think the answer's somewhere in between. I really do, because you still need someone to make that commitment, and often that is a company. Any chemistry questions? Anything you've always wanted to know? No? Good. Go and ask that guy. That's great.
Well, it's interesting, that whole thing about the virtual money that you're always promised to, and how hard it is to defend that sort of stuff, because it's a real pitfall, and it's a long-term sort of strategy, but somebody's got to hang in there while you're trying to do the innovations. So true. You hear so much about that, whether it's even sometimes worth patenting something, and given what you have to do to defend it and actually extract money from it. Yeah,
I mean, we got the 30 million up front, which was a lot of money at the time. Not that we were working for a CRI at that stage, so we didn't see much of that. But it certainly helped IRL out at the time, because I think they were having financial difficulties, so that sunk into a hole, and so it can be done. The 530 million, I don't think we tapped into much of those dollars, I have to say. But it's a difficult. I mean, it's a high risk process, and that's another question the government has to ask itself. What is the risk appetite when it comes to this sort of investment? If you want a high wage, low emissions economy, these are the sorts of businesses we need.
I think in our hearts, after watching so many movies, we'd like to believe that a discreet suitcase was slipped silently under your desk, that you open it up and there were little goodies inside, but I don't think it works that way.
No, unfortunately not. No. Maybe it's still in my office, I'll have to go and have a look.
Anyhow, thank you very much for that.
Thanks, Steve.
Presentation.